On this podcast, you'll find interviews with high-performing, successful individuals in life sciences. On a weekly basis, we cover their proven methods, principles, strategies, and mindsets to implement new technologies that scale to meet the needs of people in our world.
Welcome to Life Science Success Podcast. My name is Don. For those of you who don't know me, I'm a consultant in life sciences. I help companies manage complexity and increase performance. Today, we're going to talk to Liang Schweizer, and we're going to talk about her company, HiFiBio. But before we get to that, just quickly wanted to mention the organizational maturity assessment that's on my website right now. For companies that are scaling and companies that are adding individuals, this is a great way to assess whether or not if your company is ready. At the end, you get a score for your organization, and you can see overall how well or how much work you actually have to do before you're ready to scale. And so with that, let me introduce my guest. In this episode of Life Science Success, we're going to be talking to Liang Schweizer. Liang is the founder and chairperson and CEO of HiFiBio Therapeutics. So welcome, Liang.
Thank you. It's nice to be here, Don.
Yeah, it's great to have you. So could you just take a moment and introduce yourself to the guests or to the audience? Sorry.
Yeah, I'd be happy to. As you mentioned, I'm the founder, chairperson, and CEO of HiFiBio Therapeutics. And personally, I'm trained as a scientist, turning into an entrepreneur. So my passion is always try to understand scientific ideas and try to apply this to make impact for the society if possible. Yeah. So I don't know whether you want me to go back to my personal history, how I get here. I'll be happy to as well.
That actually would be great. Yeah, I mean, I think it's always great whenever we can hear sort of the story of what led you to the moment of founding HiFiBio as well. Yeah.
Yeah. So I grew up in China originally. So during my early years, science is always a fascination for me. Right after college, in 1989, that is, I came to the US right away to study bioprocessing engineering. That was one of the leading field at that time to combine biology and engineering together. So I entered this program at the University of Minnesota. It's one of the pilot program in the country. And that time, I'm probably leading the world as well, to try to think about how to tackle biology using technology. So we're building one of the earliest flow cytometry at that time. So since then, after I graduated, I found out, the industry and the society are not ready yet for getting into this interdisciplinary field. So I started going back to biology because my undergrad was biology. So I got my PhD actually from University of Zurich. So I moved to Switzerland.
Oh, wow.
That's where my Schweizer coming from. My husband is Swiss. So I decided to, getting back to biology, study cell signaling. And that's another fascinating field where I see, cell signaling plays a multiple role from early development, for any organism to diseases in human. When signal dysregulated, it becomes diseases. So after PhD, I went to Sloan Kettering Cancer Center, did my postdoc with Dr. Harold Bamas, Nobel laureate. At that time, I got exposed to cancer patient directly because the hospital is right next to the research institute, as well as get to see all this world-leading cancer research, got a lot of exposure for advanced ideas. Every one of you know the earlier immunotherapy. Jim Allison at that time also was at Sloan Kettering. So from a lot of exciting new ideas, I started to think about, instead of doing academic research, I should using those ideas, getting to more applied way to get into industry. So I joined Bristol Myers Squibb early on. That's my first job after my postdoc, and I stayed there 12 years and starting how to develop cancer drugs initially to broaden my area, getting to multiple disease areas and leading a group of scientists trying to, discover drugs as well as optimize it all the way, bring it to clinical trials. So during that time, I also got exposed to biologics, that is antibody therapy, at that moment when we start in BMS develop immunotherapy. We were one of the leading, companies trying to develop immune modulating antibodies for treating patients with cancer, and that was big success. And since then, I've been just fascinated about that, and with my job moving on to head of Asia cancer research, to later on in the entrepreneurial world, HiFiBio is the second company I got involved in as an entrepreneur. I've been always stayed in antibody therapy, immune modulation. That's where HiFiBio is. And going back to my roots, where also, apply technology into scientific problems. So HiFiBio, we're using single cell, a very unique technology, with interdisciplinary collaborations from 5 scientific co-founders, 3 from Boston area, 2 from Paris, putting ideas from physics, biology, antibody, and genomics all together. So that just basically, the reason I wanted to explain my past history is it explains why HiFiBio as HiFiBio it is. It's very interdisciplinary, cutting edge science, and I'm always trying to figure out solutions for patients.
Yeah, whenever I've researched this and looked at your website and things like that, it's just absolutely fascinating. And this idea of having multiple disciplines come together, under one roof is, just a fascinating thing as well.
Yeah. So one of the things I try to describe those, interdisciplinary thinking, novel thinking. I find out it's really hard to describe. So I said, okay, maybe let me invent a name. So we trademarked the name called Drug Intelligence Science, DIS. So in my company, everybody knows what DIS means, and I hope soon enough the whole world knows what DIS means. So what we trying to DIS is trying to looking at science and drug and intelligence, because using single cell, you generate easily millions of information at the single cell level within hours. and how do you really understand those large amount of data? You need a lot of machine learning, AI type of approaches, but data itself cannot really, help the patients. So you need to understand the underlying scientific foundations, and you come up with scientific hypothesis. At the end of the day, you want to develop good drugs, link the science and all those intelligence together. So that's what we're doing, drug intelligence science. and once again, highlights the power of always getting people with expertise from different areas together. So in our company, there's immunologists, there's data scientists, there's machine learning experts, there's engineers who actually build our single cell microfluid systems. People really understand physics, including imaging, fluids, and all sorts of disciplines. So it's a very exciting place to work.
Yeah, I know that fluidics can be a big part of, a lot of the work that we do, but at the same time, I don't typically hear it on the treatment side. So just curious overall. So if you were to look at, there are a lot of therapies that are coming that are like CAR T cell therapies or NK therapies that are coming. I'm not a scientist, so, you may have to explain this a little bit. How is it different than those other therapies? How is it that what you're working on is different?
Yeah, so definitely what you described, different modalities of the drug. which can all be very effective for its specific needs. And when there are certain situations, small molecule works better, certain situation, antibody works better. There are times gene therapy, cell therapy is very effective. And so those different modality, all, it's necessary. But for us, trying to understand patient, why the disease happens, will give you insight which modality is the best way to do. So when you're looking at any disease, is very heterogeneous. So the area I'm most familiar with is cancer. So if you look at tumor microenvironment, there's different stroma cells, immune cells, even within immune cell, as you mentioned, there's T cells, NK cells, macrophages. So there are so many different type of heterogeneous way. So sometimes you're looking at this disease, you don't know what tool to use to treat that disease, right? Different modalities, different tools. So single cell provides you the science you need at that resolution. So if you're looking at a mixture of cells, you still don't know what tool to use. But if you look at single cell, you say, oh, I know, in this tumor microenvironment, the really bad player is the NK cells. So, the bad player is one of those T cells got exhausted, and let's reactivate that. So you understand the disease mechanism from a single cell level. So that helps you position your drug into the right patients. And you also could use that to understand biomarkers. So how do you stratify patients? That's also a big question industry is facing. So using our DIS, we try to get to help the patients to figure out what are the best drugs to use. In addition, we are developing the drugs ourselves. So when we develop the drug, we're not just thinking about a situation. We already start, understanding during the development of the drug, understanding where it's positioned as the best. So we're combining, using DIS, trying to find the relevant target, the best modulator of a drug, and putting the right disease context. So that's what we try to achieve here.
Yeah, it's very fascinating to me because so I've done a fair amount of work in next generation sequencing with a couple different companies now. And with those companies, you can see in their data that the patient, a true cancer patient, their DNA just isn't sort of restructuring itself, appropriately. The ATCG doesn't sort of match up anymore in a lot of the cancer cells. And so, now all of a sudden, having companies that are understanding this more and sort of applying therapeutics becomes more and more important overall. So it sounds fascinating to me. And like I said, I'm not the scientist, but whenever I see that, it's like, why would, being a human, right, you would think that our bodies sort of only work one way, but no, all of a sudden, once you've mistreated it enough and let's say your T cells are exhausted, then,
Yes.
I can understand now, that there may need to be some sort of way to react to that.
Yeah, absolutely, Don. And one of the things I also wanted to highlight, right, in industry, we benefit a lot from early on the genomics era where people start doing sequencing, and later on the proteomic era, right? People say, okay, gene is not enough. I want to see how protein do. But what you're looking at is only a snapshot. So you have to look at the cell level where there's the gene, there's the protein, and there's the spatial temporal elements into those biology. So in a live cell, that's what we're looking at. Where the gene is, when the gene expressed what protein and where they're going to be expressed. Are they in the secreted or on the surface or intracellular? And those are linked to the biology. So I think, in my mind, the next wave from genomics, proteomics should be single cell biology. That's the reason in industry attracts more and more attention at the single cell level. And just add to that, early genomics, when you get a lot of information, sequence information, but what you often get is a bulk sequencing. That means any events that is not frequent, it can easily get buried or lost during that sequence. So when we do sequencing at the single cell level, you don't miss any of those rare events, situations where things just get averaged out. Yeah.
Interesting. Yeah. Um, so where is HiFiBio at today? Do you have direct clients today, or are you still, early stage?
So this is a great question. So ever since I founded HiFiBio Therapeutics in July 2017, it's time goes really fast. It's already past 5 years. So during that past 5 years, we identified 8 clinical candidates. Right now, we have 3 immuno-oncology drugs in clinic, or wait to be in clinic soon. And we also out licensing 2 other immuno-oncology antibody therapeutic candidates. So we still have a few potential clinical candidates looking for partnership or waiting for opportunity to bring it to the clinical trials ourselves. So this is, seeing the drug intelligence science making its impact using our own internal pipeline efforts. But we are also happy to share this type of approach, this type of ideas with industry. So that's the reason why we set up a partnership. In addition to the drug itself, as I mentioned, to have out license, we also using the platform, single cell platform, to collaborate. Previously public announced, we work with Takeda, Kite, and others, helping them either looking from the antibody therapeutic area or cell therapy in the situation of Kite, and also using our single cell. Recently, we're working with a Kite for novel target discovery. So we are eager and happy to share this kind of cutting edge technology or novel approach to industry. So we're welcome to work with other players, in the field, can benefit from a single cell precision for the patients.
Yeah. So it sounds to me like other pharma companies could reach out to you, and especially since they could be, listening to the show here, maybe we should talk a little bit about, what it's like to reach out to you and get started as well then. So do people normally go to your website and then contact you, and then they could be at any stage with investigating a single cell candidate, or,
Yes.
How does it work?
Yeah, excellent question. So our website is the easiest one to contact. There's a contact button and we respond, trust me. So the other thing we actually also actively working on is just, to go to conferences. JP Morgan is coming up, and we're meeting a lot of players there to show them what we can do. And so they can contact us at single cell level. They can, explore target discovery. They can explore, how to, identify a great therapeutics through single cell technology. They can also, more importantly, I think a lot of translational applications can be done at the clinical trial setting, right? So before you go to trials, we have already so far accumulated around 9 million single cell transcriptome, covers 16 tumor types and 8 autoimmune diseases. So we have a portal we actually generate ourselves called Select, where we putting public curated information together with our proprietary single cell information together, and using machine learning to make sure all this data can integrate seamlessly, making one big story. And you could explore, whether your target of interest in what disease area, what specific type of tumors or autoimmune disease make most sense, whether the mechanism associated with this target is really there. For example, our front runner, TNFR2 and OX40 agonists, when we decide to go to clinical trials, we're not going all-comer approach. We looked at our database. We find out indications where those targets are, more prominent expressed than other indications. We know it's through T cell activation in a situation of TNFR2, also through NK. So we look at those biology, is the right T cells associated with the target there in those tumor microenvironment. So we use, just intelligence approach to narrow it down to the type of indication we want to choose. Further, then we getting those samples from patients. We can do exploratory work like ex vivo. We treating those immune cells from patients with our drug ex vivo to see whether they're responding or not responding. For example, whether specific T cell clonal types would expand, proliferate upon our drugs. It's really fascinating. Not every T cells will respond to your drug, as you could imagine, but there are certain ones respond to your drug better than others. So you choose the ones that respond to your drug, and upon the responding, what are the downstream consequences of the whole entire transcriptome? So you gather those information, you do machine learning, then they tell you potentially predictive biomarkers. Then you use that in your clinical trials. You try to validate in phase 1. Then once upon validation, you go to phase 2 and 3, choosing the right patients. So in that way, imagine the clinical trial doesn't have to have thousands of people to get the statistic significance, and your response rate could be enriched. Of course, you cannot treat every patient with the same indication, but you select a subpopulation where the response rate is no longer today's 5-10%, for example. You could easily get to 30% or even above 50%. This is just our inspiration, and we try to get this new ideas validated in a clinical setting. If that can be done, it's going to be a game changer. I hope people will develop drug differently afterwards. Yeah.
Yeah. I mean, for sure, it absolutely sounds like fascinating place to be, and I certainly would encourage anybody that's listening as well to just reach out to Liang and the team at HiFiBio on their website. So Liang, there are 3 questions that I like to ask every guest. What inspires you?
I think the basic answer is curiosity. That's how it brought me to science. The second thing is compassion. I really always love to help people. That really makes me feel making a difference for the society. And the third thing is really you wanted to make a impact. I think that's more final personal satisfaction in the end. Personally, you feel you achieve something when you make a big social impact.
Yeah, my level of curiosity always, I mean, it's what led me to this, to putting together the podcast. It was, sort of, talking to people and understanding more about what they were doing and trying to find a mechanism to do that more. Right. And so here we are,
Yeah.
but I agree with you on the, curiosity on the scientific front, which I don't feel like I can have as much of an impact on, but I can certainly, help in a lot of other ways. But yeah, I look forward to seeing what HiFiBio can do with that. I have a second question for you. What concerns you?
Oh, there's so many things concerns me in today's world, right? So as we just came past, COVID-19 pandemic, and there's the preparedness of us as human society for treating, diseases like that, and also just diseases like cancer, for example. We know this for a long time. To this day, we still did not have a good solution, right? My mother passed away last year due to cancer, and I could not figure out an effective approach for her. So that was heartbroken. And So from that perspective, anything from disease to war to, political tension, all those things really makes me feel all of us should be working together to find solution for a better world.
Yeah, I agree with that. And thank you for sharing that. My last question is, what excites you?
Oh my God, there's so many things excites me. Any progresses. I remember, last year when we got a really great results and somebody showed me, I said, my hair stand up on my arms. That's so exciting. It just really, any discovery and that discovery not only linked to the science to daily work. I also love to travel. And I've been to more than 50 countries, and I love to climb mountain. Last year, I went to the top of Kilimanjaro. It's a personal dream of mine. Many, many years ago, when I was a child, I was always dreaming about get to the top of Kilimanjaro, and I did it last year. So that was a very exciting moment as well.
How long did it take you to climb Kilimanjaro?
I choose the 9-day route. I know there are people who have 7 days. I took a longer route. The success rate was higher, but also this one stretch is notoriously more challenging. And basically towards the end, you have a little vertical climb. So that was really exciting.
Yeah, I think there's a picture of you somewhere. It's either on LinkedIn or on your website. And you have your arms outstretched and things, and you're at the top. I saw that, but I was like, yeah, you never know sort of the journey that people have taken to try and get there, and it certainly is quite an achievement as well.
Yeah, it's super exciting. I also went to Mount Everest base camp. You probably see it now. That was my post always each time I get to the top of somewhere.
Yeah.
Hopefully we can post that for HiFiBio as well.
Yeah,
One of the days maybe.
I live in Colorado, and so I climbed my first 14er this last year, and it was amazing to see, but at the same time, as you get closer and closer to the top, at least here, it's more and more treacherous. And somebody had said to me that that they wouldn't climb mountains where there are knife edges, and I had no idea that that there was going to be like a rock face, on some mountains that, just as that peak that you got to sort of straddle to get, to the very top. And I was like, oh my, but yeah, I mean, Everest or Kilimanjaro are certainly well out of my league as well.
I think just being nature, that's the best thing could happen to anyone. Just,
yep.
Be out there, be active. And I think Colorado is an amazing place to be. I lived in Switzerland. You could imagine every summer we go out to the mountains.
Yeah. Well, thank you so much, Liang, for being here and telling us about HiFiBio. I greatly appreciate you being on the Life Science Success Podcast. Thanks a lot.
Thank you very much for giving me this opportunity. Have a great day, Don.
Absolutely.
Happy New Year.
You too.
Thank you for listening to Life Science Success. For complete details about this podcast, including show notes, how to get in touch with guests, and more episodes, please visit www.lifesciencesuccess.com. If there's someone you'd like for us to invite to the show as a guest, please let me know by sending me a message at the podcast website. Please click subscribe on your favorite podcast app, share the podcast, or tell a friend about it, and last but not least, rate the podcast. Thank you again.